Good morning, everybody. Thank you for coming to the very last, and as Annie and I also view as the most important session of the conference. And it is my great pleasure to introduce the speaker for our keynote today. I'm sorry. Okay. Sylvia Richardson is professor of biostatistics at the University of Cambridge and was the director of MRC Biostatistics Unit from 2012 to 2021. Before joining University of Cambridge, she held the chair of biostatistics in the Department of Epidemiology and Biostatistics at Imperial College London since 2000, and was formerly director of research at French National Institute for Medical Research, INSERM, where she held research positions for 20 years. In 2019, Sylvia was awarded Commander of the Most Excellent Order, British Empire, CBE. I'm deeply impressed. For her services to medical statistics. Sylvia was also being awarded the Guy Medal in Silver from the Royal Statistics Society and the Royal Society Wolfson Research Merit Award. She was elected to the Academy of Medical Sciences in 2016. She is a fellow of the Institute of Mathematical Statistics, IMS, that's us, and ISBA and ISI. In 2017, she received an honorary doctoral degree from University of Oslo. Sylvia has worked extensively in many areas of biostatistics research and made important contributions to the statistical modeling of complex biomedical data, in particular from Bayesian perspectives. Her work has contributed to progress in epidemiological understanding and has covered spatial modeling and disease mapping, measurement error problems, mixture and clustering models, as well as integrative analysis of observational data from different sources. Her recent research has focused on modeling and analysis of large data problems, such as those arising in genomics. Since January 2021, Sylvia has been the president of the Royal Statistics Society, the 5th woman to become president in the society's 187 years history. Sylvia also founded and co-chaired RSS COVID-19 task force, which engaged a network of RSS members during this pandemic. I have to say, I was so impressed and in awe when I was reading about Sylvia. And if I continue reading it, I will take up all the important time for her talk. So without further ado, I'm going to make the long story short. Sylvia is an inspiring person, and it's a gift to our profession. Please join me in welcoming Sylvia, please.
Thank you very much, Regina, for this very Warm introduction. Since this is the last talk of the conference, and I have a really privilege and it's a great honor to have been asked to give this talk, I think I want to take this opportunity to say first how much I've enjoyed this week, and I'm sure of seeing the discussions and all the sessions, how much you've all enjoyed it. And I think it's opportune now for us to thank Regina and Annie and the local organization committee and the whole scientific committee for having given such a fantastic start to this new series of IMS conferences, and we know that the next one is in Lisbon. So I think, could you please join me in So now to the topic of today. As you've seen from the title, I'm going to try to talk about scalability of Bayesian modeling in order to increase impact on real-world problem of our statistical models. And the health context has changed very rapidly in the last, say, 10 years. In two directions, more or less simultaneously. We're collecting much more data on vast number of patients, and particularly with their large electronic database of records. We've heard of the talks mentioning there, the large cohorts. We've also got new technology to probe much more in depth the biological mechanism. So we're also collecting a lot of joint data on individual to try to understand, the biological mechanism behind disease. Also, as we've seen over the last few years, we sometimes require to do things really in a hurry, and we've had talks talking about, how to speed up algorithm, how to take shortcuts to actually being able to deliver that at pace. And that's some of the work that I've done with some colleagues. We had led us to think of that agility, developing agility in the framework of how we think about building model for large systems, because now, with all the challenges which are actually facing us in the world, there's a lot of really large systems analysis which needs to be done. And the contribution of statistics is really to increase agility and adaptability of the systems. And so, what I wanted to discuss today is a range of a few different areas of work where either we have tried to deal with the large N framework or with the large P framework. First, for Bayesian analysis, the large N is a particular difficulty because, if you try to use, we've been used to build really nice, complex model, trying to capture the essence of what we know about the data, about the science behind. But these models are typically very slow to run. The standard type of MCMC, which has been really fueling, the impact of Bayesian inference, sort of grinds a bit to a halt when N is very large. And so in order to counterbalance that, divide and conquer strategy, people have thought where we can use some decomposition. If you are after a posterior, you could try to split your data into bits and then analyze each bit separately, and then in an informed way, in a mathematically correct way, pull things together. Now, so this works really well for a fixed model, and which has a fixed number of parameters, but it is harder if you think to want to have more kind of complex questions, like if you want to do model comparison in that way, and I'll discuss that, or if you are even dealing with models which are essentially of variable size because their complexity is linked to the number of samples. So this is an area I'll discuss. And then in terms of the large P, lots and lots of covariates. In fact, it's lots of covariates, but they are sometimes predictors, they're sometimes responses, because you want to kind of follow the path of the biology. And so what is interesting now is to try to find the interplay between these layers with smallish samples. These are expensive data to collect. So smallish sample, a few hundred, rarely thousands. But when you've got tens of thousands or possibly responses, responses or covariance. So I will discuss some, the way we have tackled this combination of kind of inspired models from the science and nevertheless scalability of computation using some optimization techniques. And then finally, just very short, because I think I will run out of time, I'll just say a few words about this business of trying to build adaptable real-time health surveillance systems. So I think I've said, so these are basically, there's two main parts, the divide and conquer, and then the scalability for high dimension in genomics. And I've got two kind of contexts within each of them. And then the last one, this is the. So in terms of divide and conquer strategy for models choice, the work was started with a PhD student, an Australian student, Daniel Afok, and then was kind of followed up by Alexander Bushel, a postdoc. So they are really the key drivers between what I'm going to talk about. And there's a paper in Bayesian analysis which just came out or something like that. So as I was sort of saying, our motivation, because I work on health data, but I just hope that, obviously not all of you works in health data, but I'm just hoping that my comments are generic enough that you could see how they could be transported in other areas where you would be dealing with, large N. Now, but our motivation was the fact that, you've got these very large biobanks and detailed genetic information, and most of the analysis involve univariate p-values. We learned a lot with that, but on the other hand, it is a bit frustrating because if you want to do even simple model comparison, you just want to do a simple model selection, it is really slow. That's the problem. So we thought, well, can we look at the marginal likelihood, which is when you use Bayesian modeling, which is a key quantity to be able to compare models, and how can we try to speed up this evaluation of marginal likelihood? And there's also cases in this particular context where you don't even have all the data in one place. You need actually distributed computing because for privacy reason or something, you're collecting data from multi-teams and each team has its own, data in a shard. So there's also an application of these types of ID when you cannot have the whole data in one place. So I'm just hearing myself an echo. So basically what we want is to calculate the marginal likelihood in order to compare models. This is the standard quantity in Bayesian inference. And these divide and conquer strategy, they go like that. You split the data into non-overlapping subsets. You run whatever you want, so your computational marginal likelihood, for example, on each of the subsets. But then you need a pooling, a principal way of putting this evidence together. And when you look at that from the marginal likelihood point of view, it is not as simple as when you just want to do posterior. If you want to reconstruct the posterior distribution of with a large N data set, and you could run, run your posterior, your just standard inference on each on the chart, there is an easy way of pulling together by this product, because you've got mathematically a decomposition of the posterior of theta given y as this product of basically this sub-posterior, and all you have to do is fractionate the price. You have to spread the information of the price. You have to fractionate it into S bits, so it takes it to the power 1 over S. And this is mathematically correct. And then you could have different way of pooling, but you've got the basis of this expression. Now, if you're trying to write this analogous kind of product decomposition for the model evidence, so the marginal likelihood, P of y there, which is the integral of that product, well, you cannot just swap the integral of the product and the product of the integral. So there isn't you cannot transpose this easy pooling to that context. In fact, you could show that there is an additional term that this is what you get if you want to use this product. that you've got a decomposition which has indeed the evidence, the marginal likelihood, which is at this middle term for each of the submodels. When I use a tilde, it means I'm using the fractionated prior. So basically it's the posterior or whatever, but it's always with this fractionated p to the theta to the 1 over s. But you're left with the need to pull together the evidence. So you're left with this integral term, this big integral of a product. And you think, well, have I done anything? I'm still left with something which is not, computable easily just by each chart separately. And intuitively, it's pretty obvious that model complexity, if you just look at if you just try to do model comparison on hundreds of your data instead of the full data set, you're going to favor a simpler model just because you've got less information. So there is a need to, again, recalibrate everything together. And this is what this last term does, this integral term. So you could compute easily the bits, the middle bit of the product, because you could use any usual way of doing by bridge sampling or important sampling to compute your, to run your posterior analysis and compute your normalizing constant. But how can you compute this integral? Now, one thing which works like usual, because the normal distribution is such a beautiful, divisible distribution, is that if you make the assumption of a normal approximation to the sub-posterior, then you can, everybody can see we can easily, because we are supposing, these splits are obviously independent, you've got none overlapping, then you could easily compute, have an explicit expression for this integral. And so that's what we proposed in our algorithm. We proposed to actually split the data into chunks, run your separate analysis, your MCMC on each of the chunks, get from that both a way of computing the normalizing constant on each of the chunks using standard, tools and program, capture the summary statistics, and particularly the mean and the covariance in each of the chunk, and then use that in an expression of this integral, and then calculate explicitly in this combined step the model evidence. And this is fast. This is fast because computing this integral becomes just an explicit expression. So this is really fast. And note that I'm using the normal approximation not for computing the evidence in each sub-posterior. You could be much finer than that, just for this last term, to approximate this last term. So in the paper, we've looked at lots of questions about the magnitude of the error that you do because of introducing, it's still an approximation of this last term, this subintegral, and seeing how it behaves as you increase the number of split or the size of the split, and look at what the practical gains in time. And I will just show you a simple example on large genetic databases. So it's UK Biobank. Here we've taken a subset that we had at our disposal, which was over 100,000 people, and split it into 20, 50, or 100 split, and then looked at regression with with Laplace priors on the coefficient, so something that you can't do that easily. You do need to run an MCMC on that, and running. So and looked at whether we could predict using either 100 genetic variants or 50. So a simple, this is like an example of a simple model choice that you might wonder, using 50 or using 100 variants, what would predict the mean red cell volume better. And we couldn't really run the full thing. So we also decided if we wanted a gold standard to what we compared to, that we would take a 10% sample, so only 12,000. And then on that, we could run the whole analysis to have a gold standard, and then again do splits to compare. So this is the graph I'm showing you now on the left-hand side here. This is only for a subset of 13,000, which we then split. So here is the actual, if you were running your MCMC and deriving your model selection so you preferred this model 1, which had 100, no, model 1, I think I had 50. You could see that basically this is preferable, but, and you could see as you're getting the subset, as they're getting smaller, some variability comes in. I forgot to say that we repeated that 20 times. So the box plot are using 20 repeat of the same mechanism, again, rerunning the MCMC change and so on. And you see the evidence of a downward bias when you get to really small size, because here this is only, each chart is now 260 items. So it is quite small. But nevertheless, the gap, stays really quite significant, and we're getting the same inference when we run on the full data set, except there we never did run long enough to know the actual truth. So this process has a downward bias, and of course it depends on the size of the slip, and it's going to be basically a tug of war between the scalability and how fast you want your results and the precision. But basically, it's incredibly much faster. We divide the computing by 200 if you use split size around 1,000 with respect to trying to do it on the full data set. So there's an error, and but it's a small downward bias, and the relative error doesn't, in this case, does not really stop the choosing the right model. So in the paper, we also do some error bounds calculations, some guarantee. It's all depend on basically the normal approximation. It's also important that the MCMC in each chart explores sufficiently so that you've got good summary statistics. I think there'll be a lot more work to be done in this area. It's an interesting area. There might be a way to improve things if you don't just do random slip, but you do targeted, stratified slips and so on. We did a few experiments, but I think there's a lot to do more and try to implement this. So now I'm going to move to my second topic on divide conquered strategy, which is connected to large-scale clustering. And this work is done together with Paul Kirk, who is a program leader in the BSU, and I've had the privilege to work with him for a number of years. And it's great. It's very, we bounce ideas together very nicely. The health context, I mean, in our health context, clustering comes again and again. We are asked clustering tasks in many times. But at the moment in the UK, there's a big context of clustering where people are interested for population health. Is they've got these, primary care databases of health record where you see a patient and are registered what is called this long-term condition, long-term, you can have diabetes, you could have the heart disease and so on, and they've got kind of codes and these records, record for each person the development and the existence of several coexisting long-term conditions. It's called comorbidity. And there's a lot of impetus in trying to move from treating a patient, one disease at a time in an uncoordinated way to try to treat groups of disease and patterns of what's called multimorbidity. So basically, we seek to use this electronic health record, which are database of millions, of patients are put together, and see to identify co-occurring patterns. So the data there is quite simple. You've got lots of rows for your patient, and then you've got columns with the conditions, and you just got 1 or 0s. And of course, you could do also longitudinal development of this, but I'm just going to use just a static description for now. And for each patient, we also record some kind of outcome. For example, has it been in that period hospitalization, or mortality or something. So you could do lots of different types of clustering. We have developed over the years something called Bayesian outcome-guided clustering, which not only, it's a semi-parametric Bayesian clustering framework, but it also uses the outcome. So you're trying to find clusters of conditions which are linked to particular, for example, mortality rate. So it's a joint clustering of the x's, which will be here, the condition, and the y associated to each cluster. There's a risk, and you try to find cluster which discriminate in terms of levels of risk as well. These clusters are typically more interpretable than if you just do fully unsupervised cluster. You discover lots of clusters, some are not particularly useful. So I don't think I've got time to go into detail. Just we've used a model that we have developed over the years for this outcome-guided clustering. Here it's in a package called Premium, and it's been used quite often. And basically, it's a joint DP clustering model, as I say, of covariance and outcome. Now, how can you scale that? The problem is that it doesn't scale. Again, if you try to run it on, 100,000 patients, it just grinds to a halt. And this problem of large-scale clustering has been recently considered in a number of papers. And again, this kind of divide and conquer type of approach, the idea you divide your data into shards, you run your clustering algorithm, and that you summarize complicated output already in each of the shards, and then you sort of match them to make up again bigger clusters. Now, this is, and there's been a couple of papers proposing, and I'm sure there are others which we're not. First thing, it's never clearly discussed whether you're using whether a fractionated prior or not is used. Nobody absolutely mentioned that. So that's one sort of question mark which we had. And secondly, we know from that, particularly when we use DP clustering, that the actual, the prior expected number of clusters is directly linked to the sample size. And in fact, on average, it's alpha being the parameter of the DP time log n over alpha. So, the model, basically, we know that if we're going to analyze a smaller data set and then pull together across this data set, we are going to get small number of clusters, and we haven't got the information to deep into, smaller substructure. So I think the target of inference, which is not really discussed of these algorithms, is not clear. What is their target of inference? This is not what I was discussing before, doing something as if you were doing the whole data set. This doesn't, in this context, this is not feasible. This is not the target that you're getting to. What you're getting, I think it's best to rephrase the problem in saying that we cannot, if we divide, reconstruct the full latent clustering we will get on the whole data set. But what we have is we can identify robust clusters that are reproducible. So we're using the fact that we have shards to see whether the structure that we're finding across the shard is reproducible, is stable in some way. And this is what we can learn by this method. But we are losing some granularity by having a smaller data set. And what we're proposing is to use basically a sort of recursing approach. So doing one pass and then doing it again on some of the large cluster, which will come up, to actually, if you want to see there's some substructure that you've actually missed, you sort of break down first pass the heterogeneity, the massive heterogeneity that you could see easily in your charts, and then you regroup your clusters, and then in each of these clusters, then you could start again and look at finer structures. So we propose first to change, to say clearly that we are not it's not achievable to do as if we were doing the whole thing in one go. And secondly, that if you are interested in finer substructure, then there's a benefit in doing several passes of this algorithm. So this is what we propose, recursing Bayesian clustering. So I think I've said what's in there. You run on each chart, you summarize the posterior draws by, to usually the posterior similarity matrices to actually have local clusters. So these clusters are called S for the shards, and K, and it's not the same number that you got. And then there's some kind of matching between the shards to get some of these global clusters. And then you could start again. You of course need to have a criteria. I just wanted to quickly show you an example to a large database of over 700,000 individuals, of which 600,000 had more than one health condition, where we actually split into nearly 600 charts of size 1,000. And we used actually premium to analyze this chart, and then we matched them using hierarchical clusters. So that's what here I'm showing you just the results over 80 shards. I can't, on the figure, you can't have the 600 shards. So I'm just showing you 80 shards where, so what we've got here is now we've done the analysis in each of the of our shards. So there you've got 80 shards. Each analysis is summarized by a certain number of each shard, some profile, and then we've clustered again this profile. So it's a cluster of clusters that you're seeing here. And here we see 4 big groups which emerges. We've just used simple hierarchical clustering. You could use whatever you want. 4 big groups which emerge. Which can be identify one, because you can't read it, but the actual, the conditions are here. There's 90 conditions or something. The conditions are here. So you see, for example, for this cluster, it's for cancer. For this cluster, it's mostly the rhythmic, heart arrhythmia. For this cluster, it's strokes and so on. So we get 4 major clusters which are reproducible across the analysis. So basically, they were detected most of the time, so in a stable way in each of the shards. And if you want to dig into deeper, you could, for example, take the 4th cluster, which is the cancer cluster, which had grouped all the clusters, and then go again on that one. And because it was only 6,000 patients there, we could just do one more run of premium, this clustering algorithm. And there we find some refined clusters among those which correspond to subgroup of cancer, and also not just subgroup of cancer, but subgroups which are associated with different patterns of comorbidity, indicating that, there are some cancers which go with different other bunch of comorbidity, which might be useful for treatment. So this is just to show you how this recursive approach could go. And I think there's a lot we're working with our colleagues, epidemiological colleagues, to actually make sense of these data, and also look at, the population public health message from those besides the technical. So to conclude this part on divide and conquer, basically, I think Bayesian non-parametric poses a challenge to divide and conquer, because the model complexity depends on the sample size. And I think by splitting, but it is useful to do this splitting, and in fact, the only thing you can do, you can never do, except distributive statistics, an analysis on 10 million, of these things. But what you can get is whether you see, you're targeting a size which you think is representative enough of something, and then you could just see if it's stable. If you get stable, basically clustering pattern across the charts, and then if you are interested in more rarer rarer clusters, then you have to do this kind of recursive approach by getting rid of the main heterogeneity patterns first and then digging deeper. So this is a practical proposal for the approach. And as I say, work is in progress with our collaborators in primary care. So now I want to switch gear and talk a little bit about how long time have I got? 25 minutes? Okay. So about scalability of Bayesian inference for high dimensional genomics. So I said already that, high dimensional genomics is where you're measuring different aspects of, not only our genetic makeup, which is the DNA, which is stable, but processes, how basically the genes act and actually gives messages to produce proteins and all the other biological products that we need to function and the cell needs to function. So we have lots of different types of biological data measured downstream, from the DNA on the same people. And the questions which are asked there is, can you do joint multivariate analysis of these data, relate one set, for example, of data set like the gene expression, which is the first kind of message passing, giving orders to the cells, what to do later on, to, for example, the way the actual DNA variants and so on. So we basically have questions. One type of integrative question is, how can we do multivariate association between large set of predictor and large set of responses? So we've got parallel responses. Another question is, we like to do network inference to really try to see what among these complex of biological or molecular data, what are the direct association? Are there quantities which are really hyperlinked, to other and which seem to be driving parts of the system and so on. So again, if we want to do Bayesian model building strategy, we use our usual two consideration in the high P context. We are focusing here not on predictive inference, but on trying to select important subsets, for example, of genetic variants, and also about a response that they're influencing. So we are it's really trying to understand the mechanism and give clues for doing further experimentation to the biology. So, we discuss with biologists to try to, the analysis are to actually give clues to, this is something, it seems that, statistically we see something going on in that area with these responses and these predictors, does it make sense? And if it does make sense, then they're going to investigate. Secondly, it's such a, you've got not huge number of samples, so you need to retain some power, because you could, if you just make very crude assumption, you find absolutely nothing. The signals are sometimes quite weak, and they're weak, and we've only got n equal to 100. So there's a limit to what you do. But what you can do is try to think about the scientific context and encode how you borrow information as best as possible. So for example, your responses, they're not independent. They might be related in some way. And so you really want to do this big hierarchical model where you share information. Similarly, when you're interested in network inference, you might want to look simultaneously at network for different biological conditions, because it's hard enough to do one network, but if you actually make some assumption that they're related in some way, then you get more power. So the two things is, thinking of sparsity priors, which are efficient but flexible, don't crush anything mechanistically, need to borrow information in a clever way. And then you need to be scalable. So you have to drop the usual MCMC algorithm and go. And then what we've done is develop variational based approaches or, EM type approaches to actually. So this work, I want to acknowledge Hélène Couffieux and Leonardo Bottolo, with whom I've developed really interesting integrative genomics modeling on what I've called highly hierarchically related sparse regressions. So as I was saying, basically, we are, the context is we've got like 20,000, typically it's about 25,000 genes. So we've got a set of 25,000 gene expression, and then we've got, so that's the types of the response sets we've got. And then we've got a fixed set of predictors, say, which are the variants. And we want to see whether some of these variants, which will be, my columns, are associated with more than one gene. If you've got a variant inside a variation of your DNA inside a gene, it's normal that, following the allele that you've got, it influences the expression. But what's really remarkable is some of these variants seem to be influencing genes miles away. I mean, in genetic miles in the genome. So they're what we call distal genes. And so these hotspots are sort of places in the genome which can influence much more than the gene. If they are in the gene where they are really of interest, because there could be key drivers of something going on, which is hard to understand, but which people are interested in. So our aim is to build these hierarchical regressions for hotspot discovery. And to be able to do that, we're going to have to borrow information in some way, because these distal associations are weak. And if you just do it one at a time and multiplicity correction, you miss most of them, basically. Simple methods just do not recover that. So, well, I don't know how much of our time. What we've done is we have done this joint, so hierarchically related regression framework. So YT is T is 1 to 25,000, my gene expression response. There's got a linear model with respect to X beta. On the beta, we're putting a spike and slab prior, usual things. And what is important, the important parameter here is the probability. So this is the slab and this is the spike. And it's basically the probability of having so it's a selection probability. And it's on this quantity that we are going to actually put our a borrowing of information model. So basically, the first stage of the model has got no borrowing of information. It's like for the moment, every regression is done separately. But at the next stage, we're actually decomposing this selection of the association between one of the response and one of the variants using a shared parameter, which is shared. So it's associated with one predictor and shared across the whole of the column. And also we have a row parameter because some genes are more variable than one. And what we have done, and I'm not going to go in the details, is we have used horseshoe priors for the interesting part is how we model, how we share. We have used horseshoe prior for looking for these hotspots. So the theta, this parameter theta s, is basically the propensity of a predictor to control a lot of things. And now you want typically You want that it controls very few things, in general, this is a rare event, so you want to actually, encourage sparsity there, but you want to allow a few of them to escape, and that's why the horseshoe is so useful there, because basically we model this theta, so this propensity shared by all the regression with the horseshoe prior, and not only that, but what's important is, I'm looking at Mark here because of there's always a scaling constant, which is really hard to, and here what we've worked in is that we're looking at the shrinkage, the overall kind of size of the shrinkage. We're linking that to the number of responses. And we in the paper, we actually do some approximate computation to show that this actually makes sense to use this particular link here. So this Q is the number of responses. So you're not going to use the same hierarchical formulation if you're analyzing 2,000 response or 25,000, because otherwise you're going to have sometimes a massive piling up effect. If you're actually fixing this and not relating to Q, you could have massive piling up effects arising. So we've developed a VB version to the MCMC. We have an MCMC version which works for small problems. It's nice because we could compare the VBL variation of Bayes algorithm to it, but it doesn't scale up to what we want to do. Here is an example where we have 20,000 responses, and we simulated 5 hotspots. So the truth is with these crosses, and we've taken true genomic regions, which has high correlation. Typically you've got these very highly correlated blocks of variants. So of course, there is, and it should be some hesitation. These two hotspots are very very linked in the genome. So of course there is some hesitation. But one trick which is necessary, because this is a problem which is well known to be multimodal, is that if you don't use an annealed variation or inference, or inspire yourself from basically MCMC tricks, you get nowhere, you get stuck. So, it's important to really try to understand what's going on in the, which we know in the MCMC framework, and then borrow whatever tricks you can to input into your optimization. So here, for this problem, where we have very correlated predictors, annealed variation over Bayes is very important, otherwise you get stuck. And this is an example which we're discussing with our colleagues on basically monocyte expression. So monocyte is a large white blood cell. It's very important for immune reaction. Experiments, they run lots of experiments stimulating them and to try to see, which how is it the control of immune system. And we've known, with the pandemic, how important, our immune system and all the perturbation it is. So we've run with 24,000 responses, 3 genomics regions of interest, which had about 15, so there's about 1,500 predictors, and 3 unstimulated, or 3 types of stimulation, and also some negative control. And for one of these regions, the region around the gene called Liz, which is a known gene, there is evidence of a hotspot, so you've got about 100, 200 out of these 24,000, which are associated with the same variant and under some different condition. So there's some really interesting, results coming up from there. And I will come back to that when I try to do a network inference of those. How long have I got now? 10 minutes? Right. So I think very briefly, I will just briefly mention that we've also used basically this horseshoe the flexibility of the horseshoe in network inference. And this is the work of Camilla Lengade, who is doing her thesis with me and Hélène Ruffieux. And we try to do scalable inference for Gaussian graphical models, but also for multiple networks. So I think we've heard lots of talks about networks, so I will go quickly there. Basically, what we want to do is look at the whole system and determine direct associations between these molecular quantities under assumption of a Gaussian graphical model, so, joint multivariate normal. And of course, you need some regularization because you're in a very large P with respect to N. Typically, you've got samples of size, 100, 200, and then you've got 500,000 possible edges or whatever. There are some, and we will want to use that on this particular example I was describing, this hotspot. Just take the set of genes which were associated with this variant and try to understand, are they mediating through one of them? What is the actual dependent structure and condition independent structure of these hotspot cluster, really. Now, there are some approaches using lasso, which are well known and have been developed for that size of problem. For example, N equals 366. We have so many, we've got altogether 380 wide genes, 5 millifiers, 300 potential edges, and ends in the order of 400 or 300. This is not, these models don't run, basically. I mean, they don't run on reasonable time. They would run, but they would take, forever. So, so what we've done is we've built on the work of a recent paper by Lee, which proposed a horseshoe prior, back on the horseshoe, but really it's fantastic as a model. And we use the horseshoe prior on the element of the precision matrix. And like usual, the horseshoe prior has got these two types of a penalty, a global scaling, and then a local scale. And of course, you put positive definite constraint and so on. But again, Lee et al. They propose Gibbs sampling, and Gibbs sampling doesn't run runs too slowly and doesn't run on large size problem. So what we've done is derived an EM type of algorithm, in fact, using a variant of EM, the expectation conditional maximization algorithm, because in this it's computationally more convenient. And we've used a reparametrization of the horseshoe, which is a classical reparametrization. And, the formulas are quite nice and explicit. And one thing which is really nice, and I was thinking of Michele's talk, is that when you're using this type of EM algorithm, when converging, when you're actually iterating, either, when there's an edge isn't present, it just goes to zero. The element of the precision matrix just crashes, and the others are clearly separated. So you don't have any thresholding and stuff that you have with the horseshoe. But one delicate question, always one delicate question, is there's the sparsity scale, the overall sparsity scale. When you use that type of algorithm, and it's been discussed in the literature in other contexts, when you're in an ultra sparse thing, it just crashes to zero. So you get nowhere. So you need to actually fix that scale. And we've used an AIC criterion to fix it by looking at increasing values of these global, scale control parameter and this AIC, and choosing the first time when it actually stabilizes, or where the sparsity stabilizes. And this criteria seems to work well to choose the tool. So we cannot maximize, we have to fix the tool. There's a massive gain of time. we can, we've also expanded that to now pull several networks together. So adding another level of the hierarchy. So now we've got the links IJ, and then you've got K networks. And what is shared between these networks is, and this latent representation is this new IJ, which is the host representation through this inverse gamma, and we just, it's this which is shared between the networks. And just to give you an example of the advantage of the performance of that, here we've got like two networks with only 40% shared edges. The estimate, so we've got two networks, one is smaller than, the N is smaller than the others. And then you've got here, the circles are the edges which are shared, and the squares and the triangles as the edges which are specific to each network. And you see this nice separation. You see that this new J parameter gives an indication whether an edge is shared or not. So when it is 0, these edges are not shared, but we are recovering network specific association. And when U is non-zero, then we're recovering the actual shared edges. So you really get a good discrimination, but you're not forcing, it's flexible enough because of the horseshoe. It's flexible to preserve the edge which are specific to each network and doesn't force them towards something. And that's what we could see some benefits of joint analysis with respect to single analysis. But more importantly, if we compare ourselves to the spike and slab joint graphical lasso, on a small sort of problem where we've got 40% of edge in agreement, you could see that the geographical lasso, it has, for the network, which is of a smaller size, the second network, it hardly recovers any specific association, whether we are recovering specific association either for the one of the network, the other, and we're recovering together. So basically this horseshoe is useful for actually preserving some flexibility to get mostly network specific, but also gain information for having when they are shared. And we have run on 10 networks. I don't have it's scalable to thousands of nodes and several networks in reasonable time. And we've done also some performance comparison with other methods, and it's on par with other methods, but there's a gain of precision because we're not forcing, we're not over shrinking between the networks. I should 4 minutes. Okay, so no, I don't have time to actually show you what we did basically. I don't have time to show you the biology. When we did this 4 stimulated condition and monocyte, we actually run some analysis and discussing those with the biologists. I just want one word about this statistical interoperabilities. I'm really changing gear. I'm now in terms of disease surveillance and what happened during the pandemic. And some of us really contributed to the statistical effort during COVID, were confronted by, there was one underlying process we're trying to understand, what's going on with infection, but there was new data set kept coming and new type of questions kept coming, and it was really, and you had to really answer things at pace. And so, we were working in a group, this very nice group, the Turing RSS partnership, which worked with the UK health security system. And so we were just trying to take we were not answering question day by day. This was the work of the data scientists of UKHSA, but we were trying just to take a little bit of a step back and think about what core issues keep coming up and where we should focus some effort on the modeling. And it led us to think about this, you know, if you really were going to design a way of doing it so that you could do things at pace and repurpose some of the model and also do evidence synthesis at pace. So a new source of data comes in, you actually want to not perturb everything you've done, but just add that information in some agile way. How could you do it? And so we came up, and there's a statistical science paper which came out about some kind of principle which we call statistical interoperability. And I don't have time to go, but if you're interested in that, I mean, modularity and being able to transfer the output from one part of your model in an efficient way, but a principal way, into as an input in another part of the model, which means that you could hang different bits of the model, but you don't have to redo the whole modeling and tweak your algorithm, or, you know, this massive mega model, which the people can't even understand which part is doing what to which part. You know, so it's kind of breaking down the problem into bits and then thinking about how you transfer information from one bit to the next in an approximate way, but principal way. And the ideas of Bayesian melding and all the work has been developed over the years about Bayesian, you know, evidence synthesis and Bayesian melding came completely into play here. And I think this is a very important area for future research. So basically, we did some work on debiasing. Combining survey data with the testing data, because we know, because people were tested because they had symptoms. If you are trying to understand the underlying prevalence, you know that you are, there's a causal link with the symptoms, so you are not, you are going to have biased inference. So, but if you have a randomized survey, and there were some in England, you actually get an anchor to get a handle on the bias. So we did quite a bit of work on that. And then after that, we fed this, we derived a sort of testing, a sort of marginalized testing data, which was, which the bias was integrated, and we could approximate it by a normal, or we could use it as sort of a point, sort of really non-parametric way. And on those, we built all the other models. So we had the kind of this core debiased prevalence, and then it was feeding into lots of other models. For example, looking at, you know, patterns following social inequality, looking at the influence of mobility and transmission, and so on. So basically, we built a little system where we had a core module there, unbiased prevalence, where we integrated basically. We created a likelihood, a sort of unbiased likelihood by integrating the bias that we were learning from randomized, and then feeding that in the other models. Right. So it was a bit ambitious as a plan, but I really wanted to have time to mention just this is quite different, because here I'm talking about agility before, and I was talking about scalability, but really agility and scalability, they go together. So I think, and I'm sure in your practice, you know, people are encountering simultaneously large P and large N, and so these challenges have to be tackled. And so I think that's really, but it doesn't mean we have to use oversimplistic model. As you've seen, we have developed quite elaborated model by combining, you know, scientific knowledge, how to borrow information, and also flexible, you know, prior who allow, you know, and I find the horseshoe is a particularly suitable prior work for borrowing information, but not pushing everything together, leaving some specificity. And in terms of scalability, then there's really a choice of approach. Some problems will be more suitable for variation-based, some for some kind of optimization for MEM and so on. But I think this kind of middle ground of we still are there, and we could collaborate with scientists to build realistic model and not oversimplistic model. And I'm not in the predictive. This is very different type of the aim of all the work we've been doing is not prediction. It's really this kind of explanatory and give clues for further work. And this combination of insightful modeling and thinking of how you borrow information with tackling the scalability is I think the way to go if we want to keep making impact in with Bayesian modeling in health and in many other areas. And I hope you could see that what I've said can be translated to probably many other areas. So first I wanted to thank all my colleagues, which I've worked, this is immense pieces of work, and I've had really the privilege to work in the MRC biostatistics during RSS Lab with really fantastic colleagues, and I hope I've done justice to all the work that they've done. And these are the references and some of the R packages, and thank you.
Thank you. Actually, this morning I was not fully awake. After reading your bio, I just realized that are we supposed to address you properly, like a commander?
Oh, no,
or because you got this.
This is just this antiquated.
No, but it's
of course not.
Yeah, it's wonderful to have this royal touch. So, any questions? Any questions?
Thanks, Sylvia, for this very impressive overview of your work. On the EQTL example, I was wondering. So you kind of split the gene and SNP effect there, right? In terms of the prior, so in terms of borrowing information. One could argue that, I mean, these things interact, and these things that you might kind of try to model in more about what you know in terms of how they interact with one another, or proximity of SNPs to one another. Would that help, or would that make sense here? So rather than doing this simple decomposition there.
I mean, I think what would help is to bring in some code data into the problem, because I mean, the actual row effect is really that some genes are much more variable than others. But I agree, we've got this conditional independence assumption given that, and you might want some clusters of some networks, and so on. So you could see that as the first pass, and what I didn't have time to show is then with the subgroups that you get, you could then try to model really their dependence. But another thing which really helps is to actually put some more information on the predictor and try to see where there's additional, effect of code data on these predictors to actually help you borrowing information. So make less, more informed, so there could be in the same pathway, there could be, some marks and so on. And so we've got a sort of version of the model which it's not exploratory. It's like when you want to refine, we see that as like the first pass, and it is indeed has this kind of a bit rough column and row assumption, but we see that as a first pass to actually see some of these joint association. And then indeed it would be interesting to either look at the network, put some of these correlation into play, or code data to get a more informed view. But this is very hard to do that large scale directly. So, so we feel it's like the next step indeed.
Okay, thanks.
Other questions? If not, please join me thanking Sylvia for a wonderful talk.